Okay, great. We're recording. So welcome everyone. This is the Neuroscience and Philosophy Salon that we have been running for a few months now. Every few weeks we have a great discussion on some interest and some really interesting topics spanning neuroscience and philosophy, and how can each inform each other and how we can actually build from the insights from these disciplines to build better philosophy and better neuroscience. So today we're going to have a discussion, a very exciting discussion, really looking forward to it. Anne Barwich is going to be leading the discussion. She is going to describe some work that she's published in the paper that I circulated. Anne is an assistant professor at Indiana University in the Bloomington campus, and she has joint appointments at cognitive science in the cognitive science program and the Department of History and Philosophy of Science and Medicine. So is that combined? Is like history, Department of History and Philosophy of Science and Medicine, right? So that's
it's a long name. So
it's a long name.
It's a long time.
Last year, as most of us, perhaps all of us here know, she published a book on how the sense of smell, using the sense of smell as a model for the mind and brain. I don't know exactly how to pronounce it, but smellosophy. What does the nose tells the mind with Harvard University Press? How do we pronounce the first word?
Smellosophy. I should say this actually started out as a complete joke. So a friend of mine doing my first postdoc went like, "Wait, you're a philosopher and you work on smell, so you're smellosopher." And the book originally, when I sent the proposal to Harvard, had a different title. It was a serious title, it was an academic title, and shortly before it came out, it was actually my editor went like, "I kind of like smellosophy."
Yeah,
so
I think
like,
it worked.
If you let me do it.
Yeah. Yes, and so Anne is going to be describing her framework, her thoughts in the first 30 minutes or so. And then we're going to have Antonella Tramacere presenting a position, discussing the presentation and the work, providing her view on it. And Antonella is a research fellow at the Department of Philosophy and Communication Studies in University of Bologna. She works on the philosophy of psychology and neuroscience, and started her research trying to understand the role of neural mirrorings in psychological development and human cognitive evolution. She's now doing both experimental and philosophical work on the neurobiology and phenomenology of self-perception. So, Anne, why don't you get started?
Okay, that's a good idea. Let me quickly share the screen. Okay. Okay, I hope everybody can see it. Meanwhile, I'm not sure how many of you actually know Father Ted, which is a British comedy series, British Irish. And I was kind of thinking about that quite often in relation to reductionism, because people quite often don't think this is a feasible position, or at least think it's an outdated one. And I'm trying to defend it here a little bit more because there are a couple of reasons, I think, why it's worthwhile rethinking what reductionism means and what we can do with it. And for today's outline, I thought I'm first of all going a little bit into the motivation I have for reviving reductionism, or a form of reductionism that I think is actually useful to look specifically at new questions in the future. So why talk about reductionism? And then, of course, I go into the main part of the talk, which is the trouble with olfaction. I'm going to address a problem in olfaction that seems to be an issue of emergence, to show that now this is actually more an issue of misconceptualization of the central causal elements, and how to rethink what are the kind of basic parts, what is their interaction in light of new technological advancements, where I focus mainly on a study. There was also this study presented in the paper that was done in the neuroscience lab by Stuart Firestein, where I spent 3 years as the kind of resident philosopher, which just 3 years that blew my mind and kind of were the time of my life. And then I go into the conclusions that hopefully will also kind of link to the discussion and Antonella's comments. So first of all, perhaps the questions, okay, what's so wrong about reductionism? It's got a very, very bad reputation, especially in the last 20 to 30 days in particular. And I hate to say it, but my former PhD supervisor was partly responsible for the decline of popularity in reductionism. So we have still kind of a little bit of a battle, a generational battle going on. And there are two quotes which kind of express the sentiment that people have against reductionism. And one is by Yek-Wan Kim, who said, being a reductionist is a little bit like being a logical positivist or a member of the old left. An aura of doctrinaire naivete hangs over him or her. So clearly, it's not necessarily the most popular one. There's the idea like this is an outdated position. How can you still defend that? Similarly, Lepore and Lobo actually said it is practically received wisdom. That always makes me skeptical, by the way, if I read something like that. What's received wisdom? Among philosophers of mind, that psychological properties, including content properties, are not identical to neurophysiological or other physical properties. And we see this position actually becoming more and more popular now, again, with the revival of panpsychism. And even though I'm not on Twitter anymore, many of you still are. So, this is a big thing right now. And philosophers like to go against each other for these kind of debates. And I think part of the way around that, and not to be stuck in this, is it mind, is it matter, is to look at the levels of organization and the way in which we're describing mental phenomena through the matter that gives rise to them. And there's a really nice quote actually by John Bickel, who described his motivation of going into the molecular and cellular basis of cognition. And since cognitive neuroscience is having enough trouble bridging a gap high in the hierarchy, there seems little reason for philosophers and cognitive scientists to attend to lower level neuroscience, except perhaps for heuristic or methodological or pragmatic reasons. And this is a good thing to rethink. So what are these kind of levels? What are these higher level phenomena? Are they really so highly organized, and lower level is just the mere mechanics of it? And when we think about reductionism, and we think about levels in particular, this is why I mentioned Dan Brooks' book actually has good kind of background for this debate. We already have a very strong assumption about the ontology behind these levels. Like, what are the key elements and how are they building on each other to create certain phenomena? This is an ontological view I don't share to explain to look, for instance, for certain explanations of what's now considered emergent phenomena, certain cognitive phenomena. I will explain what I mean with that a little bit in a moment. But perhaps to be partly polemic and partly actually supportive of the problems with reductionism is there's been a variety of really, really good works criticizing naive accounts of reductionism. As I mentioned before, some were, for instance, presented by my former supervisor, John Dupre, in The Disorder of Things, where the second big part of the book is on reductionism. And they gave it, like anti-reductionists, gave a couple of really good reasons why it's not so simple. And in particular, biology has a lot of kind of complexities involved that make an easy categorization and delineation of parts partly impossible. So context is a key part of these arguments. That being said, based on these criticisms, we're now finding many papers who say, well, biology is so complex, it's interrelated, intertwined, entrenched, entangled. It's hyper interrelated, complex. Whatever that means, at some point I wonder, what does that actually mean? It's became kind of a self-running prophecy. And if we look at the arguments, it's mainly against privileging structure over function, for instance, where it said like, well, reductions focus too much on structure explaining specificity, and we need to look at context, for instance. So rather than parts, for instance, we need to look at how we basically can map out a network of spatial and temporal relations rather than just focusing on the parts. I agree, but I don't think this is necessarily an issue against reductionism. Then the kind of old school strawman reductionism that's often kind of evoked here, as I mentioned just now, are that we've got these kind of levels of organization that are hierarchically organized. It's a bottom up from genes to behavior. We need to find bridge principles and so on. And that, as I mentioned, function rather is determined by context, not the parts. We've got also very often a functional promiscuity and a top-down regulation. I do think these points are absolutely valid, but I don't think they're necessarily a good or sufficient reason to abandon reductionism, but rather a good motivation to rethink when reductionism is actually a valid strategy, and helps us also to understand these kind of contextual complexities. And a wonderful quote I find to think about what's happening here, especially in modern neuroscience, is to look at the change in methodology. And there's a paper by Dan Nicholson, which got a lot of attention in 2019 and also last year, where it was mainly against the machine metaphor, but in relation to that, he also argues quite often against mechanistic explanations. And he gave a quote that made my ears raise, because I think it encapsulates very nicely what's at stake here and why this debate still matters. Because he wrote at some point that single molecule studies are yielding results not anticipated by the use of population averaged methods. These results are bringing about a radical shift in how we think about the cell, replacing a mechanical, neatly ordered, rigid picture with one that is inherently stochastic, more plastic, and less predictable. What we are witnessing, in effect, is a conceptual revolution being triggered by a methodological revolution. And that kind of made my ears go up, because I think this is kind of what also, I do think there's definitely a conceptual revolution based on a methodological revolution, but I think it's a revolution that should make us embrace reductionism in a modern form much more, rather than abandoning it. So a change in technology, we're going away from rigid state, rigid structures, to some kind of dynamical modeling. We're looking more at probabilistic state spaces. Is that really the end of reductionism, or even causal mechanistic explanations? And I think not. So I need to give a little bit of a background to what extent we might have abandoned reductionism as a good explanation, especially the differentiation between structure and function in certain studies. And the reason for that, I think, is historical. And since, well, history at some point kind of develops, it's good sometimes to revisit it and see what's still applicable and what's not. And the person in the center here is David Marr. And there was an argument also presented by John Bickler in a 2015 paper. And when I read the first chapter of David Marr, which is basically thrown at everyone loving neuroscience, I found it fascinating, because he gives this beautiful account of the history of neuroscience when it comes to the kind of development of studies of the visual system. And when it stopped being revolutionary at some point, where there was a stagnation, and there's a really kind of revealing quote, namely, after all these kind of, but somewhere underneath, something was going on. The initial discoveries of the 1950s and 1960s, so we've got Hubel and Wiesel and all these kind of revolutionary developments, were not being followed by equally dramatic discoveries in the 1970s. No neurophysiologist had recorded new and clear high-level correlates of perception. There seems to be at some point more and more, especially after that kind of revolutionary book, a split between computational neuroscience and bench neuroscience. And too often the bench neuroscientists dealing with their proteins were kind of seen as a little bit of a handmaiden to the computational neuroscientists to do the great models, the great theories, and the rest is just data. And Pickle basically made a very good argument in saying, well, okay, back then he had a point, because back then, if we do look at single cell recordings, we couldn't necessarily get a broader picture of the function. This is what the idea of the computational theory in Marr was like. Okay, the single cell recordings don't give us an explanation of in what kind of function and organization they're involved. But that's not so much applicable anymore, because that changed with the introduction of genetic intervention techniques that gave us a way to do direct manipulations and interventions to look at causal mechanistic explanations. So new technologies, in effect, are more than just interventions as a basis of explanation and providing more data, but they're also becoming more and more a source of genuine theorizing. So tools as theories, which I recently read a really nice paper by Gerd Gigerenzer, who actually made a similar account of tools as theories. And I think there's a very good point looking at that, what that also means for reductionism. And when we look at these really, really cool, I gotta say, I mean, the brain is just sexy when we look at all these kind of new imaging techniques. It's the most photogenic organ. And so, but beyond that, beyond just the sex appeal, so to speak, there's also a really nice realization of, because that just shows us two things, this kind of methodological, technological revolution. First of all, cellular mechanisms no longer provide just mere details if we try to go and get at the kind of higher level computational models and the physiological processes. But I do think they also constitute the material foundation from which to derive better theoretical models and better theories in neuroscience. And the failure of a lot of previous reductionist explanations and previous reductionist models can quite often be shown actually to be a misconceptualization of the key causal element involved. So one thing is when we say we can't reduce X to Y, well, these are not theory neutral elements. What do we consider as the parts, the building blocks and their interaction? There's already a lot of theory here. And this theory, I think, hinges on the way we've been investigating these materials and their technologies. And this is where I finally come to olfaction. And I should warn you, feel free to at some point go like, okay, don't speed up, but stop, because I can talk about smell the whole day. But my favorite part about olfaction is that it was often dismissed as being pure subjective, you know, and I think my whole mantra is I want to remove the stain of subjectivity from smell, which isn't actually as hard as it might sound. But there was one quote by Leslie Kay, who's a neuroscientist at Chicago, and she gave a talk at the Association for Chemoreception Sciences in 2018. Fantastic talk, and at some point she got lots of questions about like, yeah, what about this confounding factor, what about that, and she gave this quote which was burning itself into my mind, because at some point like, yeah, I get it, this is confounding, this kind of varies the results, there's a lot of kind of variation here, but if everything is contextual, what remains the same? And I think this is one of the most important questions to ask, not just simply about olfaction, but olfaction as a model system for many biological phenomena, because variation is not subjectivity. Variation is often elicited by some underlying mechanisms that are the shared principles we're trying to understand. So that question got me into the rabbit hole of looking at the kind of commonalities between these different variations in smell. And smell first seems to be actually the best case against reductionism or some form of surface reductionism, because we don't have a straightforward stimulus response model, unlike vision, unlike audition, because we can't just simply reduce the odor of odorants to their molecular structure. But that's not an issue of subjectivity. There's a lot of complexity involved. So the question is like, how can we get at certain commonalities dealing with that complexity? Smell has extremely irregular structure-odor relations. And as an example, for instance, I gave you 4 different musk molecules here on the left side. Now, musk is one of the key ingredients in perfumery. It's one of the most well-studied type of odorants. And you see there is no structural commonality. I mean, you have completely different configurations. You even have atom groups, like, for instance, nitrogen here, which others don't share. In fact, you have an constantly expanding repertoire of odorants, because galaxolide, which you see here, was synthesized and invented, so to speak, in 2012. Smell is the sense where you're constantly actually adding new structures. So that's just as an example, like why it's so difficult to figure out find a way around the chemical complexity. Furthermore, it's a matter of really big data, because there was a study in I think it was 2014 or 15, that said, well, humans can discriminate up to 1 trillion odor stimuli. There was a bit of debate around that number. I think the official number right now is just shit tons, frankly. So it's growing. There's a lot of complexity. Most minute nuances can lead to differentiations in perception. And that's not even where it stops, because we're talking here about single molecules, but we hardly smell monomolecular mixtures unless you're in a lab and an experimenter goes, smell this or smell that. Quite often we're exposed to hundreds and hundreds of odorants, not just in our environment, but coffee has several hundreds of odorants. So we perceive mixtures, and mixtures are tricky. They behave unpredictably. You just need to ask perfumers for that. So this is a very long quote, but Christophe Laudamiel, for instance, he said, well, if you have a mixture and you've got your formula and you're trying to, for instance, play with a formula and twist and tweak it, you've got 2 phenomena. The first one is sometimes, you know, you have a certain solution and you're adding a little bit more in, just a tiny minute amount, and it unpredictable changes the entire configuration, like the configural perception of the mixture. And sometimes you add something, nothing. You add more, nothing, not a single change. So there is clearly the phenomenon that, so to speak, as Aristotle said, the whole is more than the sum of its parts. My favorite example to illustrate that is actually the perfume Eternity by Calvin Klein, which has the ingredient of indole in it. And indole in and of its own smells of shit. It has a very strong fecal scent, but you don't smell it, of course, in the perfume. Otherwise, it wouldn't have been such a big success. Kind of what my fun part about that is that eternity actually has shit in it. So this is, I don't know, maybe this is kind of perfumery humor, but that just as an understanding of how complex mixtures are and that they behave unpredictably, because sometimes you add stuff, nothing happens. There seems to be some perceptual suppression effect, or vice versa. You've got a complete configural change, a form of what is in there by the most minute changes. So it's unpredictable. And you see that with food odorants as well. Sometimes you can create from very, very few key odorants a complex image that's not reducible to its parts, so to speak. An example is potato chips, another study by a colleague of mine at Cornell, Terry Akery and his lab. And he showed that to what extent potato chips, the perceptual image of potato chips, can be created by three key odorants, which in and of their own actually do not smell of potato chips, but potato, toast, and rotten cabbage. The interesting thing is that it's the ratio, not just simply the set, but the ratio in which these odorants come together that create this nice perceptual picture. So something is going on here with mixture perception, which I find quite fascinating. And I think the answer is not chemistry. Quite often you see that people then try to find the kind of chemical features that explain that, but it's biology. And it's not higher level biology. It's not something that the mind simply creates. I think it's really on the molecular level. So, excuse me. This is why I think we need to deep black box biology, at least in olfaction, and unfortunately in many other areas, not all, of course. Quite often biology is treated as this black box, because we can just have the stimulus and the perception. So the question is there sufficient reason to think that knowledge of receptors and their behavior affects and changes our understanding of odor coding? The short answer is yes. The long answer is heck yes. So I won't give you the long answer, but as I said, I could otherwise talk about it the whole day. But quickly about the receptors, they are fascinating. But there hasn't been as much insight into the olfactory receptors compared with other protein receptors, partly because they're fairly young. They were only discovered in 1991 by Linda Buck and Richard Axel, who showed that olfactory receptors are actually part of a superfamily of G protein-coupled receptors, which are a highly diverse family, lots of evolutionary conserved amino acid secret motifs, and also they're the largest multi-gene family in most mammalian genomes, so dolphins are an exception. They occupy about 4%, just the olfactory GPCRs of the mammalian genomes, and GPCRs in general about 10%. So it's huge. Humans have basically about 400, sometimes you read also 300 something, or 420 members in humans, at 1,000 mice, 2,000 elephants. So that should cover the complexity part I've been talking about, to just say, we need to kind of think about what's going on here. The problem is that complexity is kind of ignored in most structure odor models. Oops, sorry, my dog just came. And what we're having here is basically this kind of ligand binding model of, well, in terms of we've got the ligands and we've got the descriptors of the things they're kind of creating in terms of the perceptual description. So we've got two data sets. We've got the chemical data and we've got the descriptors. In between are the receptors, and they're somehow never treated when we're doing structure-odor relations, as if we can do a stimulus response model and understanding of smell without dealing with the receptors that select features in order for the brain to read them off the receptor sheet, so to speak. That's not even what we do in vision. Why do we do that actually in smell? Something I always found quite puzzling, because there's a third list. A third list in the sense of, do the receptors really respond to the same features we're actually modeling from an analytic chemist perspective? That's an empirical question that was actually addressed only recently in 2017 and 18, again by my former Freistein lab. And they showed to what extent they did an approach with medicinal chemistry, looking at how the receptors, or in this case, the olfactory sensory neurons, which express each like one receptor type, respond to these different odorants. And they used a set of ketones. And in the second study, they also used esters, which have kind of a fruity smell. Turns out that the receptors couldn't care less about how an analytic chemist would arrange these molecules as more similar or not similar. You get a completely different similarity tree in this context. So the difference between these biological explanations and the chemical explanations, I find are eye-opening, because their divergent already indicates something which piqued my interest, namely that the real mistake in previous models are we can't reduce odor to molecular features and to chemical models, is that the stimulus response models excluded the receptors. We operated with the wrong causal elements. So we need to rethink what's going on here. And the failure of previous reductionist models does not mean that it's necessarily something in the mind or emergent, but we're overlooking something here. And just to give you some kind of idea to what extent they're really diverging, if you ask analytic chemists like, okay, can you classify these ketones for me? They would go like, well, first of all, we separate them by the size of their ring. Then we go for the composition of the ring. Then we go for the subfamily. Do we have a nitrogen, an oxygen, and sulfur atom, and so on? So there's a kind of catalog of chemical similarity. But chemical similarity is not a given, because the receptors, first of all, don't care about ring size whatsoever. Neither does actually the cycle composition. What they're really, really keen on is the polar surface area. And the question of like, will the neuron accept the odorant as a ligand? The fun part is, I don't have that here, but there was a study where they threw a machine learning algorithm at a bunch of, like a lot of collection of a data set in 2017, a science study. They haven't even mentioned polar surface area. So this is why we really need biology beyond the kind of computational tools we're having. I think there needs to be a bit more interaction between these different techniques and to take biology a bit more serious on its own account. And that's, again, just single molecules, but I'm getting more and more obsessed with the question about mixtures, because I think we need to model olfaction from the perception of mixtures rather than single molecules and then trying to find more and more complexity by adding molecules, but rather the other way around. So it's kind of a paradigm shift. And the classic mechanism, what was considered to be the mechanism of odor coding in individual, like in monomolecular mixtures and complex mixtures, was combinatorial coding, mainly that you've got one receptor that can respond to different parts of different odorants, and one odorant can be detected by different receptors via different parts. So this is why you get this massive combinatorial explosion of possibilities that you can smell. This might not be the only mechanism by which we're operating here, and that is basically the study I kind of presented in that paper, which is the SCAPE study, I often want to mention. And SCAPE stands for, okay, let me breathe in first. Swept confocally aligned planar excitation. Say that 5 times in a row. This is what I think people do approbations. Which is it was invented in 2015 by Elizabeth Hillman at Columbia University, and it's a hybrid between light sheet and confocal scanning microscopy, and it allows you to actually scan entire living organisms if they're small enough, like fruit flies or larvae. It's pretty cool, and you can also have large tissue, intact tissue parts of larger organisms, such as mice, for instance. So, and it's fast, it has a high resolution, and it allows you to scan a lot of cells at the same time. And that, of course, suddenly provide an opportunity to study mixtures. Maybe because finally you could actually throw a mixture at the receptor sheet and record a large amount of cells responding to a mixture, something that wasn't possible before. And if you're interested in how people made that methodology work, so Lu Shu, she's the lead author of that SCAPE study, and we wrote a paper together looking actually at her challenges in making that method work, this new technology for this area, which I found quite fascinating to see what she struggled with, how certain reasonings come. So if you're interested in how to make technology work for a new problem, Lu gave a first-hand account, which is coming out soon. So when it comes to the mechanism of odor coding, they showed some really, really interesting new findings. And I will come back to how that deals with reductionism in a moment. But they recorded about 10,000 cells, which is quite a lot compared to previous studies in olfaction, but where they had basically, they used genetically modified mice, GCAMP 6F mice, and showed to what extent there was up to 38% of responses to a mixture that was different to the kind of mixtures with individual odorants. So they had 2 mixtures of each 3 odorants, and they consulted Christophe Lodamiel, the perfumer I just mentioned earlier, in terms of what would be actually a good a good sample to use when it comes to chemically and perceptually dissimilar odorants, and they tested one in equal and the other in unequal concentration. What was really interesting is that they had two effects. One was a suppression effect that cells responded up to 30% less, which is a lot, and also enhancement effects that they had up to 30% suddenly responding to something, to a tripart mixture, without responding necessarily, cells not responding to the individual parts. It was a long question, but okay, suppression was kind of a bit expected, let's put it that way, because you sometimes have some kind of antagonistic relations, but enhancement, what would that mean? Like, that is kind of an interesting discovery. So what they discovered actually was, first of all, that there's a mixture dependent agonism and antagonism, and some are concentration effects, but not all, meaning basically that it's not the odorant that necessarily has particular functional features, but the odorant in a mixture in relation to the receptor. So it's a relational properties. And the other thing is they discovered allosteric effects sorry, my dog is super excited right now. Allosteric effects in terms of, well, there might be an enhancement and a way in which the receptors work that so far haven't been recorded on any other sensory periphery. And I'll come to that in a moment. So this is basically just to show you the setup where you've got the perfusion chamber with large swaths of the nasal epithelium, and from that they could then record a really nice volumetric data of the epithelium. Now, just need to show you the picture because I think it's kind of a neural firework. I mean, sometimes some data slides are just nice for aesthetic effects, and this is one of them. Let me actually show to you the response to one of the mixtures on a selected area of the epithelium. Unfortunately, it doesn't have sound effect. I was kind of hoping that they might at some point do kind of a nice sound layover, but whoops, that's just, okay. So first of all, when it comes to the suppression effect, the interesting thing is they could, through that kind of single cell recording, show which one was actually the inhibitor and which one was not. So if you look at, for instance, the case here, the isoalderane clearly was kind of seen as the inhibiting factor, because they could do not just simply the tripartite mixture, they could compare it with a kind of 2 mixtures, like in one case with dartanol and isoalderane, and to show to what extent there is definitely one of the 2 that acts as the inhibitor. So it's not just necessarily the whole mixture, like every odorant having the same responsibility or the same functional relevance within the mixture. Suppression, as I said, was kind of a little bit expected. So there were kind of parallel studies pointing at something similar, and it wouldn't be so surprising to have that in comparison with other protein studies. What was surprising is, and this is where I'm kind of rushing through a little bit, was the enhancement effect. Namely, that you actually have sometimes fewer cells responding to the, I have to just quickly put you to the side because, okay, what you have here, for instance, is that you have fewer cells responding, more cells responding to the entire mixture in comparison to the individual odorants. So you've got an enhancement effect. So how can that be that more cells are responding to the mixture than to the, let's say, addition or the kind of sum of the cells responding to individual odorants? And that was something that was for a long time, so it took a while to get the study out, puzzling both Stuart as well as Lou. And I kid you not, this is one of those pure accidents. 1.5 days before I had to submit my book, got an email from Stuart saying, I think we might have an explanation. Lou came up with a model that would explain it. So if you have to change an entire book chapter 1.5 days prior to it coming out, I didn't have enough coffee in like that much coffee in a long time. The proposal is actually not a not so unknown mechanism. And Lou told me, like, well, the moment you mention it, people are like, it's obvious, but for some reason nobody thought of it. Namely, perhaps it's allosteric interaction, meaning, and this is kind of a simplified version, but okay, you might have an odorant to which the receptor is not responding. But if you have an odorant in the mixture and there's a second odorant that then would interact with basically the hypothetical, sometimes some words have got the German and the English word and they do a tango, with the hypothetical allosteric side, changing the configuration of the receptor such that it's then interacts with the other odorant. And you might wonder, okay, but what's the point of that? What's going on here? Well, this is a model that would explain a lot of puzzlement when it comes to mixture perception. I mentioned combinatorial coding, and I mentioned that lots of mixtures have hundreds and hundreds of odorants. So if you have combinatorial coding and you've got this swath amount of receptors responding, how does the brain know what the nose is smelling? Because at some point the signal gets so complex and smears out, but we're very good at discriminating different mixtures. We can even train ourselves to discriminate different kinds of wine. So if there's so much receptor activation, at some point, there wouldn't be any specificity anymore. So what we're having here is actually a form of distinguishing the signal from this kind of smart amount of combinatorial coding. It can either lead to differentiating signals when it comes to enhancement of two similar mixture and also a sparsification of the signal. This is basically an answer to the question of how the brain deals with mixtures to still be able to discriminate between otherwise too much data being combinatorially coded. So with combinatorial activation alone, receptor activation patterns quickly overlap to form a broad and smudged signal, which would lose its distinctiveness. Modulation, antagonistic, and allosteric that facilitate a unique receptor code for the discrimination of complex mixtures. This is why I think this is a study that really has revolutionary implications for how in the future we need to think about olfaction, because so far we've been mainly dealing with single odorant studies. That was a pragmatic choice, but it has shaped our understanding of olfaction so much that we now need to rethink what's the basis, what are the causal elements, etc. And I think this is a study that allows us to do that, because what it basically shows is to what extent a function is indeed determined by the context. So you've got mixture-dependent antagonism and antagonism as well as enhancement. It also shows a certain functional promiscuity, keeping the specificity. So I'm going now through the different pieces that were thrown against reductionism. So we've got different binding sites for different effects and modulations, and showing just to what extent really receptor biology has gone a long, long way since the original lock and key stuff, which in olfaction was first proposed in 1946, actually by none other than Linus Pauling. We have something that explains a seemingly emergent psychological effect. We've got a configural perceptual quality recorded in perfumery, as I mentioned earlier, that actually arises from mixture coding patterns as determined by receptor modulation. So it further prompts us to rethink the basis of objectivity and subjectivity, because it's not just simply the input-output axis, but the determinant elements are basically the coding mechanisms. That also means we need to think about what's involved in that. Is it the individual receptors, or is it a certain population behavior? And how is that actually more a relational property rather than just an issue of individual elements? So what Mark considered as the computational level is determined by, and I think must be derived from, the implementation level, not the other way around. So this is the key message I want to highlight. I do think that quite often we're underestimating the specificity of the biological materials that we're dealing with. And that brings me to the last bit, where basically, I think the theoretical analysis of empirical phenomena, it is always contingent on the way in which we study the materials. So the methodologies that underpin our understanding of elements, the levels and interactions. This is why I also earlier highlighted Dan's book, for instance, because what are the levels? What are the elements? This is methodology and technology dependent. This is not a given necessarily. Quite often we rely on certain understanding, but sometimes it needs to be shifted. New technologies, like for instance, the escape study, have I think, a real potential to overthrow some of our kind of preconceived notions and understanding. And this is where we need to see to what extent some of understandings are built on certain technologies that have become so deeply entrenched in our methods, and how new methodologies might overthrow that. We have that phenomenon in many biological issues. Why did mice and fruit flies take over? Well, because they fitted really nicely with some genetic techniques. We lost a lot of model organisms, and the theoretical loss that comes with it, of course, we should keep in mind. So this is where, so like, we need to think about the causal levels, whether we talk about cells populations, and to what extent context actually can be explained and determined and looked at as a mechanism if we think about what the elements are and how they're reacting. So what actually constitutes the principal elements and interactions. So new tools, I think, allow identifying the mismatch between the natural conditions and the laboratory model in the system's function quite often. Because as I mentioned, the laboratory condition in olfaction was single molecules, while in reality we study mixtures, and this is where we need to rethink biology. And that is basically I think it was the last figure in the paper, where it shows us to what extent we really rethink the ontology. Why this is a reductionism. Reductionism not in sort of, well, we just go all the way down to our previous ontological model, but more, what are the elements? It's maybe we can eliminate some of the mechanisms that were inserted as an explanatory tool to explain certain effects that become unnecessary once we shift what the causal elements and their interaction are. So it's a form of reduction on a methodological level as well as on an ontological level. And with that, I want to thank the lab that made it actually possible. So Stuart Feuerstein is here, and Lu, who's the lead author of that study, is here, as well as Erwan, Namin, and Dongjing. And with that, again, thank you for actually listening to my very, very long rant, but I kind of get excited about olfaction. That makes it hard to stop.
Thanks so much. That was wonderful. Okay, so we're ready to hear your thoughts, Antonella, before we open more broadly for discussion.
Okay, I'm going to share the slides. And are you seeing them?
Oh, perfect. I love the brain.
Thanks. I found it on the internet. So thanks for inviting me here. And as I said before, I'm very happy to see Anne again, and Luiz, that I also only had the possibility to see on Twitter, and all of you that are here joining the discussion. So these are my comments on Anne's paper, Imaging the Living Brain, and how the Imaging the Living Brain makes, according to Anne, an argument for ruthless reductionism from olfactory neurobiology. I will proceed in this way. I will first very, very briefly recap the key points of our article, and then I will pass on discussing what I like about the article and what I like a little bit less, or at least what left me with some questions that I would like to address or to open up for discussions later. So what the article does is to defend reductionism in neuroscience, and not simply a general type of reductionism, but especially in particularly explanatory reductionism, to be distinguished from theory reductionism, where we are not interested in finding logical principles to connect the theory holding at an higher level to the principles of the theory at a lower level. And so she defends this kind of bottom-up theorizing, where the interest lies in finding or seeing whether and how representation of at a higher level could be linked back, explained from representation, or at least the causal behavior of lower level entities or variables. And she shows in this paper that in some cases, or at least in this case, emergent properties or attribution of emergent properties result from the misconceptualization of the key elements that play a causal role in the phenomenon that we are observing. And she does that by discussing these new and actually disregarded technologies that regard escape microscopy and how it is applied to olfactory research. So what I like about the research, many things. The paper is very engaging, is very nice. It is an exciting topic, and especially it treats something that has been a topic that has been disregarded for so long in cognitive science, and as a consequence in philosophy of neuroscience and mind. Also, I really like that she discussed this, the article discussed